Today this will be discussed about erythropoiesis. Its components include introduction, definition, stage of erythropoiesis, factors necessary for erythropoiesis, factors necessary for hemoglobin formation. First, discuss about introduction. Bone marrow is the tissue in your bones that is the factory for producing blood cells. Red blood cell formation is the process called erythropoiesis. Red blood cells carry oxygen and transport whole body. Red blood cells transport of nutrition also. About 1% of RBC are replaced daily. Normal lifespan of RBC 100 to 120 days. Second definition. Erythropoiesis is the process in which origin, development and maturation of red blood cells. Stage of erythropoiesis. First stage pro-normoblast, second early normoblast, third intermediate normoblast, fourth late normoblast, fifth reticulocyte, last matured RBC or erythrocyte. Diagram for erythropoiesis. First, bone marrow produce pluripotent stem cell, also called HSG, hematopoietic stem cells. It produces proerythroblast stage, next stage early normoblast, continue stage intermediate normoblast, late normoblast, reticulocyte and matured RBC. Stage of erythropoiesis. First pro-normoblast. Pro-normoblast is the first stage cell derived from CFU E, colony forming unit E. It is very large in size with a diameter of about 20 micrometer. The nucleus has 2 or more nuclei and a reticular network. The cytoplasm basophilic in nature. Second stage early normoblast. It is little smaller than proerythroblast with a diameter of about 15 micrometer. Nuclei disappear in nucleus. The cytoplasm is basophilic in nature, so this cell is also called basophilic erythroblast. Next stage intermediate normoblast. The cell is smaller than the early normoblast with a diameter 10 to 12 micrometer. The nucleus is still present. The hemoglobin starts appearing because the presence of hemoglobin it stands with both acidic as well as basic stains. So this cell is called polychromatic erythroblast. Late normoblast stage. Late normoblast stage size is about 8 to 10 micrometer. Nucleus become very small quantity of hemoglobin increase and cytoplasm become almost acidophilic. Next stage reticulocyte. Reticulocyte is also known as immature red blood cells. Size slightly larger than mature RBC. The cytoplasm contains the reticular network. Due to the presence of reticular network, the cell is called reticulocyte. It stands with supravital stain. Normal value of reticulocyte: adult 0 to 2%, newborn baby 2 to 6%. Matured erythrocyte. Reticular network disappear and the cell become the matured RBC and morphology biconcave cell shape. The cell decrease in size 7.2 micrometer in diameter. The matured RBC contain hemoglobin but without nucleus. It requires 7 days for the development and maturation of RBC. From pro-erythroblast take 5 days up to reticulocyte stage. Reticulocyte takes 2 more days become to matured RBC. Factors necessary for erythropoiesis. As the demand for new cells production, the bone marrow requires large amount of hemoglobin. Development and maturation of erythrocyte require various types of factors. First, erythropoietin, thyroxine, hemopoietic growth factors, vitamins. These are the factors necessary for the formation of red blood cells. Next, factors necessary for hemoglobin formation. First, class proteins and amino acids. Protein of high biological value are essential for the formation of hemoglobin. Amino acids derived from these proteins are required for the synthesis of proteins part of hemoglobin. Protein globin. Second, iron. Iron necessary for the formation of heme part of the hemoglobin. Third, copper necessary for the absorption of iron from the gastrointestinal tract. Fourth, cobalt and nickel. These metals are essential for the utilization of iron during hemoglobin formation. Fifth, vitamins. Vitamin C, riboflavin, nicotinic acid and pyridoxine are also essential for the formation of hemoglobin. Thank you everyone.